Hi, I'm Katie Krupnick, and for my exam mini video number 4, I used nail polish to illustrate the process of transcription in prokaryotes. This medium felt particularly fitting because it is my 1-year anniversary of COVID, during which time I have become quite the do-it-yourself nail artist. We'll begin with the core enzyme, RNA polymerase, which binds with the sigma factor to create a holoenzyme, which we use in transcription. The process of transcription has 3 main stages: initiation, elongation, and termination. We'll start at initiation, which is where DNA begins the process of unwinding when our RNA polymerase holoenzyme binds to the promoter region. The promoter region is made up of the -35 box, aka the control region, the -10 box, aka the Tata box, and the +1 box, which is the transcription start site. It should be noted that the -35 and the -10 box regions are also called consensus sequences, which are present in the segments of interest and are recognized by the sigma unit when bonding. Here we have elongation, which begins after the DNA strands are separated into a transcription bubble. The transcription bubble, which can be seen here, is where the RNA polymerase moves across the template strand in the 5' to 3' direction, creating RNTPs. Remember, the DNA strands are anti-parallel, meaning that they are opposite from each other. Also, elongation uses just the core enzyme after the sigma dissociates after 9 to 12 base pairs in order to produce RNA transcripts from left to right. Finally, we have termination, which has 2 main pathways: the row-dependent or direct repeat, which uses helicase to unwind the DNA-RNA hybrid, thus ending transcription, or row-independent or inverted repeat, which leads to hairpin loop formation in RNA, which signals the end of transcription. Hairpin loops are followed by a string of adenine called poly A tails, which allows for dissociation of the RNA strand. Finally, to address the question from the prompt, if 5 base pairs were inserted between the -35 box and the -10 box regions for a gene which codes for phenylalanine, we would encounter issues because the base pairs which initiate transcription would be pushed out of the Tata box, and DNA polymerase would then not be able to bind and would not be recognized by sigma factors. Therefore, we would not even start transcription for the phenylalanine, and it would not be present in the organism. Thank you so much for watching my video. And finally, here are my references.